Welcome back to BioBits and Bytes for another deep dive. Today we're going deep into the world of DNA and RNA sequencing, focusing on nanopore sequencing. For those of you who have been following advances in biological understanding and research, you've probably heard about all kinds of different sequencing technologies. And today we're going to break down nanopore sequencing and what makes it different, without getting too technical.
Right.
We'll start with the basics of DNA and RNA, and then jump into how nanopore technology reads these molecules.
Okay.
And finally dive into some of the real-world applications that are becoming possible thanks to this technology.
Sound good?
So let's start with the fundamentals.
Sure.
DNA, the blueprint of life. RNA, the messenger.
Yep.
And proteins, the workhorses of the cell.
Exactly.
So this flow of information from DNA to RNA to protein is the central dogma of molecular biology.
Yeah, it's like the foundation of how our cells work.
Okay, so DNA uses that four-letter alphabet.
You got it.
A, T, G, and C.
Right.
And RNA is similar, but swaps out the T for a U. These bases aren't just arranged randomly, are they?
No, not at all. There's a very specific pairing. It's like a lock and key system. Adenine, or A, always pairs with thymine or T in DNA. And in RNA, it pairs with uracil, or U.
I see.
And then guanine, or G, always pairs with cytosine, or C.
Okay.
This is called complementary base pairing. And it's what allows DNA to replicate and be transcribed into RNA.
It's amazing how these simple pairings make up the code for all life.
Absolutely.
So the sequence of these letters, the bases, is what we call the genetic code, right?
Precisely.
And this code dictates the construction of proteins.
That's right.
But how does that translation from code to protein actually work?
So in messenger RNA, or mRNA, the sequence of these nucleotides is read in groups of 3.
Okay.
Each group of 3 is called a codon.
A codon.
Yes. And most of these codons specify a particular amino acid,
Got it.
which are the building blocks of proteins.
So it's like a 3-letter word.
Yeah, you could think of it that way.
That translates to a specific building block.
Exactly. And there are 64 possible codons,
Wow.
but only 20 common amino acids. So this means that multiple codons can code for the same amino acid.
Oh, so there's some redundancy built in.
Yeah, we call it degeneracy. It's like having synonyms in a language.
Huh, that makes sense.
Different codons can mean the same thing.
Are there any codons that don't code for amino acids?
Yes, there are. There are start codons and stop codons.
Oh, okay, like punctuation marks.
Exactly.
So tell me about those.
The most common start codon is AUG in RNA,
AUG. Okay.
which signals the beginning of protein synthesis.
Like the first word in a sentence.
Right. And AUG also codes for the amino acid methionine.
So it does double duty.
It does. And then we have the stop codons.
The periods at the end of the sentence.
Yeah, like UAA, UAG, and UGA in RNA.
Okay.
These tell the protein-making machinery to stop adding amino acids and release the completed protein.
Makes sense.
What's amazing is that this genetic code, this way of reading codons, it's nearly universal across all forms of life on Earth.
Wow, that's incredible.
It suggests that this is a very ancient and fundamental mechanism for life.
Really puts things in perspective.
It does.
So we've covered DNA, RNA, and the genetic code.
Yep.
Now let's talk about how we actually read this code.
Sure.
How do we figure out the precise sequence of these letters?
Right.
That's where sequencing technologies come in. And we're focusing on nanopore sequencing today, but I think it's helpful to understand the context.
Sure.
So can you tell us a little bit about traditional methods like Sanger sequencing and Illumina sequencing?
Absolutely. So Sanger sequencing was the first widely used method. It's based on selectively incorporating chain-terminating nucleotides during DNA replication and then separating these fragments by size to determine the sequence.
And what about Illumina sequencing?
Illumina is a much newer technology,
Yeah.
and it's become very popular because it can sequence millions or even billions of DNA fragments simultaneously.
Wow, that's a lot.
It is.
So how does it work?
It uses a technique called sequencing by synthesis, where DNA fragments are first amplified, and then fluorescently labeled nucleotides are added one by one. As each nucleotide is incorporated, a laser excites the fluorescent tag, and the color of the light emitted tells us which base was added.
So it's like reading a colorful code.
Kind of, yeah.
Yeah.
It's a very powerful method for sequencing relatively short stretches of DNA very accurately.
So it's all about accuracy with Illumina.
Yes, and high throughput.
So how is nanopore sequencing different?
It's a fundamentally different approach.
Yeah.
It's based on passing a single strand of DNA, or RNA,
Okay.
through a tiny nanopore.
A nanopore, what is that exactly?
A nanopore is a tiny hole. It's a nanometer in scale.
So incredibly small.
Extremely small, yes.
Yeah.
And this nanopore is embedded in an electrically resistant membrane. in a device called a flow cell.
Okay.
Think of it like this. The membrane is like a wall with a tiny hole in it.
I see.
And this hole, the nanopore, is what the DNA or RNA has to squeeze through.
Interesting.
So each nanopore has an electrode,
Okay.
which is connected to a sensor chip. And this chip measures the electrical current flowing through the pore.
Makes sense.
Now, when a DNA or RNA molecule passes through this nanopore, it causes a disruption in the electrical current.
So like blocking the flow?
Yeah, you could think of it that way.
Yeah.
And what's really amazing is that each of the 4 bases, A, T, U, G, and C, they all have slightly different shapes and chemical properties.
Okay.
So as each base passes through the nanopore, it creates a characteristic change in the current.
So each base has a unique electrical signature.
Exactly.
That's incredible.
It is.
So we're not looking at colors or anything like that.
No, it's all about detecting these subtle changes in electrical current.
And this is all happening in real time, right?
Yes, as the DNA or RNA is moving through the pore, we're getting the sequence data.
So it's like watching a movie of the DNA being read.
You could say that.
So this is very different from Illumina, which takes a while to process the data.
It's much faster.
So can you walk us through some of the advantages of nanopore sequencing?
Sure. So one of the biggest advantages is the ability to generate ultra-long reads. Yes. While Illumina typically produces reads of a few hundred base pairs, nanopore can sequence fragments that are hundreds of thousands or even millions of bases long.
Wow, that's a huge difference.
It is. And these ultra-long reads are incredibly valuable for assembling complex genomes.
Why is that?
Because they allow us to span repetitive regions and structural variations
I see.
that are very difficult to resolve with short reads.
Makes sense.
Another big advantage is real-time analysis, because the sequencing happens as the molecule is passing through the pore.
Right.
We get the data almost instantaneously.
So it's like live streaming.
Yeah, exactly.
Yeah.
This is really useful for things like rapid pathogen identification during outbreaks
Okay.
or making real-time adjustments to experiments.
So speed is a key advantage.
Absolutely.
And I understand that nanopore sequencing can also directly analyze native DNA and RNA.
Yes, that's another important advantage. We don't need to amplify the DNA using PCR.
Which can introduce bias.
That's right.
So nanopore provides a more direct view of the genetic material.
It does.
And this also allows for the detection of base modifications like methylation,
You got it.
which are important for understanding gene regulation.
They are.
So we've got long reads, real-time analysis, direct sequencing. What else?
Well, nanopore technology is also very scalable and portable.
Okay.
There are small portable devices like the Minion,
Indian.
which can be used in the field for environmental sampling or point-of-care diagnostics.
Wow, so sequencing is no longer limited to the lab.
Exactly.
That's amazing.
And there are also larger high-throughput devices like the Promethean for large-scale sequencing projects.
So it can handle both small and large jobs?
We can.
That's impressive. But I imagine that dealing with those tiny electrical signals must be pretty complex.
It is. The raw signal from the nanopore sequencer is quite noisy.
Noisy.
Yeah, there's a lot of background noise that needs to be filtered out.
Yeah.
And the signal itself needs to be interpreted.
So how do we make sense of all that data?
That's where artificial intelligence, or AI, comes in.
AI.
Yeah. We use sophisticated algorithms called base callers.
Base callers.
Yes. To translate those electrical signals into a sequence of A, T, G, and C.
So the AI is essentially reading the squiggles.
Yeah, you could say that the AI is trained on massive data sets of known DNA and RNA sequences and their corresponding nanopore signals.
So it learns to recognize the patterns.
Exactly. And then it can apply that knowledge to new data.
And this AI is getting better all the time, right?
It is. The accuracy and speed of base calling are constantly improving.
That's great to hear.
And if we ever start sequencing truly non-standard nucleotides, the role of AI will become even more critical.
I see.
Because the algorithms will need to be trained to recognize these new signals.
So AI is essential for making sense of the data.
Absolutely.
So with all these advantages, what are some of the real-world applications of nanopore sequencing?
The applications are really diverse and growing rapidly. One major area is genome assembly. The long reads from nanopore sequencing make it much easier to assemble complex genomes, especially for organisms with lots of repetitive DNA.
Like what?
Plants and some animals. They often have very complex genomes. And nanopore can help us get a more complete picture of their genetic makeup.
I see.
Another area is the analysis of structural variations, which are large-scale changes in the genome, like insertions, deletions, inversions, and translocations. These variations can play a big role in disease, and nanopore's long reads are really good at detecting them.
So it can help us understand the genetic basis of diseases.
Exactly.
And what about RNA sequencing?
Nanopore is also having a big impact on RNA sequencing, or transcriptomics. It can sequence full-length RNA molecules,
Okay.
which gives us a more complete view of gene expression. We can see different isoforms and splice variants, which are important for understanding how genes are regulated.
And when we talked about epigenetics earlier,
Right.
how does nanopore help with that?
It allows us to detect base modifications like methylation directly from the sequencing data, so we can study how methylation patterns change in different cell types or in response to environmental factors.
So it's providing new insights into gene regulation. And I understand that nanopore is also being used for things like microbiome analysis.
Yes, the portable nature of the minion makes it ideal for studying microbial communities
Yeah.
in the field, like in soil or water or even in the human gut.
So we can learn more about the complex ecosystems that microbes live in.
Exactly.
And what about infectious disease?
Nanopore is also being used for rapid pathogen identification, which is critical for outbreak response. Imagine being able to quickly identify a new virus or bacteria. During an outbreak,
That could be a game changer.
it could. And it can also help us track the spread of antibiotic resistance.
So it's a very powerful tool for public health.
It is.
And while Illumina is still the gold standard for clinical diagnostics,
Right.
nanopore is finding its niche in applications where long reads or rapid turnaround time are important.
Yes, for example, it's being used for cancer diagnostics and for identifying genetic disorders.
So it's becoming increasingly relevant in medicine,
It is.
and we're also seeing it being used for environmental research
Yeah.
and conservation efforts.
Yes. For example, it can be used to monitor biodiversity and to track endangered species.
So it's a versatile technology with a wide range of applications.
It really is.
And it's constantly evolving. The
Well,
accuracy is improving,
Yeah.
and new applications are being developed all the time. It's really an exciting time to be in the field of genomics.
It is.
So as we wrap up this deep dive, I'm struck by the transformative potential of nanopore sequencing. The ability to generate ultra-long reads, perform real-time analysis, and directly detect base modifications is really opening up new possibilities for understanding the code of life.
Absolutely.
And it's exciting to think about where this technology will take us in the future.
It is. Imagine being able to sequence the entire genome of a cancer patient to identify the specific mutations driving their disease, or rapidly identifying new pathogens to prevent future pandemics, or even gaining a deeper understanding of the genetic basis of complex diseases like Alzheimer's.
Those are some really big goals.
They are, but I think nanopore sequencing has the potential to help us achieve them.
It's really a powerful reminder of how quickly our ability to decipher the code of life is advancing.
It is.
And I encourage all of our listeners to keep exploring this fascinating field. There's so much more to learn. And who knows what amazing discoveries are just around the corner.
Exactly.
Thank you for joining us on this deep dive into nanopore sequencing. Until next time, keep that curiosity alive.